Okay, so I'm going to be presenting the Case 8 case study about MHC Class 2 deficiency. First, just to provide a brief summary about the structure and major functions of MHC Class 2. We've already covered these thoroughly in class, but the study did talk about them at the beginning. And the purpose of an MHC molecule is to bind peptide fragments derived from pathogens and display them on the cell surface for recognition by the appropriate T cells. And there are two classes, MHC1 and MHC class 2. MHC class 2, specifically talked about in this study, is involved in presenting antigens to CD4 T cells or T cells that have a co-receptor on their surface, CD4. The peptide antigens that MHC2 molecules present are derived from extracellular pathogens and proteins taken up into intracellular vesicles. And MHC2 molecules are expressed on antigen-presenting cells, generally dendritic cells, macrophages, and B cells. And you can see from the figure on the right how already activated T cells that have bound to whose CD4 molecules have bound to a peptide antigen are now carrying out their effector functions on a macrophage and then also on a B cell. They are heterodimers that are highly polymorphic, and they consist of an alpha chain and a beta chain. And for this study, it's important to know that they're encoded from genes located on the HLA locus on the short arm of the chromosome 6. And on the figure to the right, it's the gene structure of the human MHC molecule. And you can see it distinguishes which regions encode for each type of MHC molecule, either class 1 or class 2. The gene expression of the alpha and beta chain must be strictly coordinated, and it's under like a very complex regulatory control by a series of transcription factors. And it's the existence and attempt to identify those transcription factors that led to the discovery and study of patients with MHC class 2 deficiency. So a brief summary of what the person that the case study spoke about was Helen Burns, a 6-month-old infant who developed pneumonia symptoms. They originally treated the pneumonia symptoms and she recovered. However, because of her pneumonia was caused by a particular opportunistic virus, they suspected that she might have some sort of immunodeficiency. And a bone marrow transplant test confirmed that. It revealed that her circulating B lymphocytes and monocytes didn't express HLA-DR molecules, confirming that MHC class 2 deficiency was the issue. Her final treatment involved a bone marrow graft transplant using her brother's matching HLA type, and that was able to successfully work and restore her immunity function. The figure to the left is showing the specific fluorescent antibody detection compared between Helen and a healthy individual. The left panels are Helen's, and they revealed that she expressed only about 1% of the amount of MHC molecules compared to a transformed B cell line from a healthy control. So moving on to the portion of the study that I'm mainly focusing on is the way they spoke about and determined the cause and definitions involving MHC class 2 deficiency. It's an inherited autosomal recessive trait, and its deficiency is identified as in having low levels of CD4 T cells. Health problems normally show up early in infancy, evidently shown by their combined immunodeficiency symptoms, things like increased susceptibility to pyrogenetic opportunistic infections, prone to severe viral infections, and moderate to severe hypogammaglobulinemia. And then something that the study also discussed, unlike other types of immunodeficiency, progressive infection of BCG virus has not been observed in MHC2 deficient patients. Because after BCG vaccination against tuberculosis, mostly because the mycobacterial antigens derived from BCG can be presented on MHC class 1 molecules. So the infected cells infected by it are going to be destroyed by CD8 presenting cytotoxic T cells instead. Moving on to the form of treatment that was used on Helen, and is most of the time the main option of treatment for patients with MHC class 2 deficiency. Although Helen was cured after a bone marrow transplant from her HLA identical brother, the results from this treatment aren't always successful. Even with an HLA identical donor, the results can be inaccurate, represented when CD4 T cells circulating still remain low. And this is thought to be caused by positive selection of donor-derived CD4 lymphocytes being compromised, meaning owing to a lack of MHC2 molecules on the surface of a patient's thymic epithelial cells. And because of this, the search for a cause was built, but it also exposed more unknown variables. One of them being that causes of MHC class 2 deficiency can range from person to person. And for example, an example that the study talked about was Epstein-Barr or EBV virus that transformed B cells isolated from class 2 deficient patients didn't express MHC class 2 molecules. So moving on to how they specifically defined the deficiency. The genetic linkage analysis in large families affected by MHC class 2 deficiency revealed that the inherited failure causing MHC class 2 deficiency is not linked to the MHC locus, and it's actually on chromosome 6. It's actually linked instead to most likely to defects that lie within the regulation and expression of the MHC class 2 genes. The gene locus on the short arm of chromosome 6 were absorbed as normal, and this meaning they were able to confirm it's not linked to the condition. Interferon gamma induces the expression of MHC2 molecules on antigen-presenting cells in healthy individuals, but it fails to induce their expression on antigen-presenting cells of patients with MHC2 deficiency, again suggesting that that the defect might lie in the regulation of the expression of MHC class 2 genes. The experiments involved in providing clues that led to the identification of the defect were pairwise fusion experiments. When B cells from two different patients are fused, MHC class 2 expression can be observed. The fusion of the two cell lines has corrected the defect, meaning that one cell must be able to replace whatever's lacking in the other, revealing that two cells must carry different genetic defects to cause MHC class 2 deficiency. The figure that they provided in the study that you see to the right, if the two cell lines do not correct each other, that's what represents the minuses, and if they are in the same complementation group and have the same genetic defect. But if the defects were corrected, that's what represents the pluses, meaning the two cell lines belong to two different defects. The pairwise functions were performed on a large number of cell lines from different patients, and then 4 complementation groups were found, A, B, C, and D. And then the 4 transcription factors that were involved were class 2 transactivator, or CIITA, RFXANK, RFX5, and RFXAP, which bind to the 5' regulatory region of the MHC2 genes. Specifically, CITA is the main factor involved here because it binds to the RFX complex that activates transcription. And then the other 3 bind directly to the promoter of all MHC class 2 genes. And it was through these experiments that they were able to identify or find clues and identify the cause of the condition. In conclusion, the lack of MHC molecules turns out to be a result from defects in the transcription factors required to regulate coordination and not in the gene locus themselves.